in this episode.
The moment, though, I started approaching, in particular, molecular genetics, which was really my first case study, I realized that if I would go in with this view of substances, substance view of the world, and see what genes are, I would have probably needed to give up on whether genes were anything, because genes are extraordinarily dynamic entities. So they are highly context dependent, so they depend on the fact that there is a cell that can transcribe them. The same gene, in the sense of the same entity that encodes for a given product, might actually be realized by different bits of DNA. So somehow genes were almost the opposite of the substance, in the sense that they actually change through change. Now, what I ask myself is like, does this mean that there are no genes, that we need to give it up, that there are no entities like that? And to me, that wasn't really a satisfactory answer, because I did think that there is something there. And normally the principle that I use philosophically for this type of situations is that I ask whether the given entity has causal power or not. So does it impact the world around it or not? And somehow with this other kind of philosophical guidance, I was like, okay, maybe genes are things, but they're just things that are a bit different from substances. They are temporal entities, so they change through time. They might come in and out of existence. They might come in and out of existence because they are in a given context. And somehow this became quite a framework in which I started thinking of biochemical entities more in general.
Hey everyone, welcome to my conversation with the self-assembly researcher and philosopher of biochemistry, Dr. Francesca Bellazzi. First of all, I should say an apology to Dr. Bellazzi, because I mispronounced her name throughout the whole podcast. I think I kept calling her Francesca, but it's actually pronounced Francesca. That's my bad. But leaving that aside, I gotta say this conversation really was going out on a limb for me, really, because I wouldn't say that I was really well read or well versed in the myriad fields that Francesca researches in. For instance, the field of biochemistry and even biology in general, apart from reading a few books by those like Richard Dawkins and, of course, my favorite, Stephen Jay Gould. I don't have any deep understanding of the contemporary research going on in these fields. However, Francesca was extremely generous and kind to even break down some of the concepts, really fundamental concepts, using a variety of examples to elucidate and make her points to a neophyte like myself or a lay audience. But what really interested me was the fact that this notion of the philosophy of biochemistry, this field, it's a very nascent and hopefully burgeoning field of study. And Francesca, at least for me, was the first person I've come across who kind of identifies as a philosopher of biochemistry, and that immediately intrigued me. And I reached out to her and we organized this conversation where we discussed a gene as a natural kind, this notion of natural kindedness in the philosophy of science. We discussed self-assembly and then the field of biochemistry in general, or the philosophy of biochemistry in general, and what concepts can be brought into biochemistry research from the philosophy of science and philosophy as a whole. Also, of course, we discussed process philosophy, which I'm deeply drawn towards, by those like Whitehead and even, you could say, in the Cornell side, those like Deleuze. So I felt there was a lot there, and I felt I could have done a better job at exploring a bit more of the technical topics of the field. However, I think I've got to just do a bit more reading on the field as a whole before delving into that. The good thing, though, is Francesca is writing a monograph on the philosophy of biochemistry. We did sort of discuss that too, and I certainly am hoping to invite her on again once that book is out to discuss the field in general and kind of dive a bit deeper, which I hope she agrees to. But before we continue, a quick PSA. You may be aware that I've mentioned multiple times I'll never have advertisers or sponsors on this podcast. I simply don't have anything to sell you, and I deeply do believe that we live in a society of excess and overconsumption, and I don't want to contribute to that by selling you more things. That being said though, forgive the tautology, we ultimately live in the world we live in, and I can't continue this project and sustain it if I don't have any kind of financial support. So as uncomfortable as this makes me feel, I sincerely would like to ask you to consider supporting this podcast financially. Needless to state, your listenership and viewership is more than enough support. However, if you have the means, please do consider supporting me on Patreon, Substack, or even PayPal. And if you're wondering what are the perks, what are you going to get? Nothing. Absolutely nothing, because I'm not selling you anything. You're not buying anything, but rather, if this podcast, if the work I've done has brought you any kind of value, if you've learned something new, do consider supporting me on these platforms, but to reiterate, only if you have the means. All right, back to the episode. As I was saying in that PSA, I would be deeply grateful if you could consider supporting me through any of these platforms. Running this podcast certainly does cost a lot of money, and especially for the academic texts that I've got to buy at times, because most times I gotta say, the publishers do send across a copy, but there are times where they don't, or they simply don't respond, and at those times I do buy the book to prepare well for these podcasts, because I want to be respectful of the fact that the guest is even making time to talk to me, and sort of enlighten me in their specific field. So any help, again, would be deeply appreciative. Okay, be that as it may, now a formal introduction to Francesca. Dr. Francesca Bellazzi is a postdoctoral researcher in the ERC-funded project AssemblingLife at the University of Oslo, working on self-assembling processes in biochemistry. Her main research explores the philosophy of biochemistry and the interface between chemistry and biology, and her thesis was titled Biochemical Kinds and the Unity of Science. She will be releasing her upcoming book, Philosophy of Biochemistry, Biochemical Kinds and Their Properties, with Cambridge University Press. With that being said, here's my conversation with Dr. Francesca Bellazzi.
I mean, just for me in general, like the kind of explicit realization that whatever I experience as a subject needs to come through space and time, that for me was just like, in a way, mind-blowing, even if it's so obvious. Like, of course, I can't think of anything around me if I don't locate them in like the present or the past or the future or in a given spatial location. But somehow that for me was like that part of the critique of pure reason. I found like really mind-blowing when I was like a young philosophy student, and I got to think through that. I mean, I read him first in high school even, so I was even a bit younger. And
I don't even know
and yes,
how you got through Kant in high school.
yeah, well, this is not my own. I can't take the merit for that. Like I'm very lucky because in Italy we start studying history of philosophy at 16. And then we studied that for 3 years. And we normally go through all the classics. And this is really how I got into philosophy in the first place. So I have quite a strong historical training first. And I remember even like as a high school student, just thinking that, yeah, oh my God, like it's true that somehow I have these sunglasses on or these lenses on, in which I don't know what there is behind those, but however I experience it is somehow coming through space and time. And that for me was, quite a strong realization that I ought Kant.
Yeah, I mean, honestly, this is what I love about European education. I know a few of my friends who are from even Germany and France and certainly Italy. The fact that even in high school, students are exposed to these classical thinkers. It's all I've been complete autodidact, like philosophy units. I don't think you even have like philosophy as a subject in Australia, if I'm not mistaken. And even in university, it's very driven towards more of like, to put it crudely, it's driven towards the fields that'll get you a high paying job. Really.
Yeah,
Yeah.
sure.
To put it crudely, it's not, but that shouldn't be the purpose of education. I mean, I'm quite critical of this kind of neoliberal model of the university. Anyway, that's a whole other. No, I don't want to open a can of worms there. But
Yeah.
Francisca, as I was saying, yeah, thank you so much for making time for this podcast for my little tiny little project. as I did tell you before we started recording, I don't think we recorded that bit. Yeah, I've never spoken to a philosopher of biochemistry. So maybe if it's okay, let's start with somewhat of a biographical note. And my first question would be, I initially thought you were a person who studied biology and then fell into philosophy, but it seems like it's the other way around, that your background all the way from your bachelor's, correct me if I'm wrong, has been in philosophy of science and your training is in philosophy, not necessarily as a biologist nor a chemist. So if you could just tell us, how did you get interested in the philosophy of biochemistry? And then perhaps through that, just tell us, yeah, what is really the philosophy of biochemistry? I mean, not like, I mean, I guess I could Google a definition, but that's boring. Tell us, like, what kind of questions do you all deal with? What do you all tackle? And, what keeps you awake at night?
Yeah, yeah, fantastic. Thank you. Yeah, so I have to admit, I am a philosopher by training. I'm not a biologist, not a biochemist. However, I need to say that I had, like, since I was really a child, this kind of interest, mostly towards genetics. And I think that I really owe that to the fact that my parents read me this book about Mendel when I was, I don't know, around 10, 11. And somehow the laws of transmission of characters like blew my mind back then. And somehow these questions regarding like trait transmission and genetics were always in the background. And actually, during high school, and when I needed to decide what to study, I was really torn between going into biology or going into philosophy. Somehow the fact that indeed I've been exposed to the history of philosophy gave me the idea that philosophy could give me the breadth that I wanted. And yeah, so I enrolled into a philosophy program, but my very first like mini piece of research was on the philosophy of genetics. So for my undergrad dissertation, which in Italy is a small project, but is already quite a substantive one, given the age. I worked on Mendel's laws. So since then, I started like looking at the interplay between basically more chemical entities like the DNA and traits and phenotypes. And somehow I carried on doing that until my PhD. When I started my PhD, it was in a project that was on the unity of science. So it's the meta science project. And my supervisor gave us a lot of freedoms, but the idea was that we needed to study some relations between sciences. Or anyway, the question is, okay, let us consider some specific instances in which we can think about the question of the unity of science, or whether sciences can be reduced, can be unified, and how. And so somehow for me, the most natural thing to do was to start with genetics. And this is how I actually then got into biochemistry, because in my very ambitious PhD proposal, I was hoping to just say like, oh, let me go give you an overview of molecular genetics and Mendelian genetics, and go through all these fascinating notions. But somehow, starting with molecular genetics, I got stuck there. And I found out that actually there are so many fascinating questions that pertain to the nature of molecular genes, the natures of DNA, and then almost naturally the nature of proteins, because proteins are the first output of gene expression. And this is how I opened the jar of the wonders of the philosophy of biochemistry. Basically, I got encouraged to study the relationship between chemistry and biology in my context, and somehow I got stuck there. Now, broadly, I take a very, I would say, broad indeed understanding of biochemistry and molecular genetics, or molecular biology in general. I know that, of course, they are different fields and they are separated, but I don't think that. I think that this is mostly due to historical reasons than theoretical reasons. I think that the questions that they study are really similar, and generally we can take both of them to study the chemical processes within and related to living organisms, or the molecular processes within and related to living organisms. Now, both molecular biology and biochemistry, I find, are quite laboratory based, and they're really proud of this. Why? Because they actually get us amazing results by using tools, knowledge, techniques that come from chemistry to solve biological problems, to intervene into biological processes. So I think that both disciplines are really kind of like laboratory practice based. And this, I think, gives us the first step to separate the philosophy of biochemistry from biochemistry, or the philosophy of molecular biology from molecular biology, because somehow we take, as philosophers, a kind of theoretical standpoint. There are very few of us that do philosophy of biochemistry or philosophy of molecular biology, but somehow, given the importance that entities such as proteins have, there are now more and more. And I would say that the questions that we study can either be from a more theoretical perspective, like asking how biochemical theories works. The first questions that come through are questions related to reductionism. So can we really describe biological processes into chemical terms? Up to which point? When do we need to start considering the context of the specific organism that we are studying? And with that, populations dynamics and principles of evolution, and when we don't need to do that. And these are really questions that come from the more, I would say, epistemic tradition of philosophy of science. But there are also questions which are really the ones that are more interesting for me personally, which is, okay, which type of entities do we find when we are there at that interface between chemistry and biology? What are the features that they present? How do they behave? Do we need to characterize them differently from chemical entities that are generally characterized in terms of structure? If I think of gold, then I would offer a periodic table characterization of the element. Like, can I think of a protein in the same way? Do I need to bring in other features? And similarly with, like, I don't know, carbohydrates, I worked on vitamins loads because I think that they give us interesting questions regarding classification. And I think that kind classification and identification is one of the big questions in philosophy of biochemistry. So how do we classify these molecules? How do we characterize them? And this can either be a very general question. So what does it make a molecule biochemical compared to chemical? But also questions that regard the identification of specific kinds, like how do we classify vitamins? How do we classify hormones? How do we classify proteins? And we might think that these questions are easy, and maybe from the external perspective, it's like, oh, obviously we know what vitamin D is. Come on, like, and in a way we do, of course we do. But in another, like vitamin D, for instance, is quite tricky because it is produced by our bodies, so it seems very akin to an hormone, but it is classified as a vitamin. And if we want to make the relevance come through, we can think that classification, like how we classify vitamin D, might impact how we want to regulate its consumption, how we want to sell them. Do you need a medical prescription to get vitamin D, as you might to get some other hormones, for instance, or you don't because it's just a nutrient? So this is just to motivate the fact that questions of classifications in the philosophy of biochemistry are really important. Other questions that we ask is, what is the function of a molecule? We tend to ascribe functions to traits. So the function of my legs is to allow me to move around. But when I say that hemoglobin has the function to carry oxygen, am I saying something that is exactly as the heart has the function to pump blood, or am I saying something different? And there are even more questions about like whether actually biochemical molecules are processes, whether biochemical contributions are just chemical reactions, or do we need to consider selected processes for them, and many other questions.
Yeah, that's a perfect example because in fact it's a good segue. I was going to ask you a bit more about self-assembling processes, but we'll get to that because you mentioned functions. I think the first paper I read of yours was this. So my memory is a bit off at the moment because I read this a while back. it's this paper, Biochemical Functions, just your paper, and then you make a really interesting point. So maybe this could be, you could use this to demonstrate more of what you meant, this question of classification. You argued that, and I took note of this, that yeah, biochemical molecules don't really have functions; they only have alterations. If I'm not mistaken, there. So, and so the notion was like, from what I understood, that you're saying that biochemical molecules don't really dysfunction in the sense they have like different variations or alterations. So I'm curious, when you introduce a concept like that, how does that differ from more of a, let's say, a conventional understanding of what a function of a molecule or even a biological organ would be?
Yeah, thank you. So I think that that point that I make in the paper is really related to the case study that I was considering, that is the presence of vitamin D, of vitamin B12, sorry, in the body. And considering the fact that vitamin B12 has a function in erythropoiesis, which is the renovation of red blood cells. Now, the point that I make there is that, well, vitamin B12 doesn't present, for instance, traits in itself because it's a molecule. And accordingly, we can't say that vitamin B12 doesn't display the traits for which it has been selected for, as we would say, for instance, for the heart, for a malfunctioning heart or a dysfunctioning heart. Why? Because it's a molecule. However, we want to say that there might be cases in which we notice something that is not going as it is supposed to be. And I think that then the discussion of dysfunctionality in these cases can either be at the level of the molecule, and this is where I say that they don't display dysfunctions in themselves. They can display alterations because, for instance, it can be a form of vitamin B12 that is a form of cobalamin, broadly speaking, that got damaged. So this is a first form of alteration, and so it doesn't play the coenzyme role that it has in erythropoiesis. Or, for instance, I might have a lack of vitamin B12, for which I don't have enough vitamin B12 that contributes to the process of erythropoiesis, or I have too much. Luckily, vitamin B12 is water soluble, so normally our body is quite good at dealing with the excess. But this can be another form of alteration. Now, this doesn't mean that there is a dysfunctionality in general. It's just that I put the dysfunctionality on the process of erythropoiesis. Now, if there is a lack or an excess of vitamin B12 or a damaged form of vitamin B12, then it is the process of erythropoiesis that doesn't do what it has been selected for, which means that it is dysfunctioning. So the view that I take for biochemical molecules in that paper, and for biochemical functions in particular, is that in order to characterize functionality, we need to consider the causal role that the molecule plays, but also the general biological evolutionary context in which this role is played. And that context is crucial for our evaluations. So it is why it is what the process of erythropoiesis has been selected for that offers us the view of functionality that we need, and the context for functionality really that we need. Now, I've changed a little bit this view, or rather, like I've kept on developing this view, because if we consider cases like proteins, then things might get more complicated, because there are some proteins for which we can identify a clear evolutionary history. And then there are some proteins that, for instance, might be like disfolded, like malfolded, or like structurally unfolded, or folded in a way that can lead to pathologies. And then maybe in the case in which we can identify a clear evolutionary history of the protein, and the protein doesn't fold in the way that it has been selected, for, then there I think I would ascribe proper dysfunctionality. But also that I don't think that we can generalize to all proteins. For instance, I do think that selection plays an important role in the identification of this type of dysfunctionality.
Sorry, I don't think I understood what you meant by like. So how has your view changed though by the
Well,
change?
well, no, maybe it hasn't really changed. I would say it has improved.
Okay, you
Yes,
developed it
yes,
more empirically, and then
yes, in the sense that I think that in order to give a kind of like meaningful and accurate discussion of biochemical functionality and dysfunctionality, we need to consider the specific type of molecule we are talking about. So in the case of vitamin B12, given that vitamin B12, it's a simpler molecule, it's not produced by the organism, so it's more difficult to identify a direct evolutionary history, then we need to make this distinction between alterations and dysfunctions and give the alterations to the molecule, the dysfunction to the process. In the case of proteins, which are the result of protein synthesis, and the result of genetic expression, then I think that there things get a bit more complicated because somehow we can have protein alteration, like if the environment changes and the protein changes shapes and this affects the functionality. We can have protein dysfunction in the case in which, for instance, there is a genetic mutation. And so the protein, the resulted protein doesn't fold or doesn't have the characteristics for which it has been selected. I think a good case in this can be the case of cystic fibrosis, in which we know that there is specific genetic mutation that causes the dysfunctionality at the higher level. And then we can still have a form of like dysfunctionality in the process to which the proteins contribute because we have a lack of those proteins or we have an excess of those proteins or because the context of the process changes.
okay, great. Yeah, now I'm just going to connect this to a bit of a speculative question here, which is the Stephen Jay Gould that idea I shared. Because one thing in your answer, one thing I picked up on is that you kept using this word context and how I really like that what you put forward, the notion that there's no dysfunctions, only alterations. I mean, I know you don't want to, use like a broad stroke paint everything in that way. However, I find that concept interesting. So the notion is, I mean, it's quite simple, really. What I'm trying to say here, it's this. I think, especially in let's say in the public consciousness, there's this very almost social Darwinist notion, which again, it's there have been periods in human history. It was in the 20th century, and I think now it's coming back again. This notion of like survival of the fittest, selecting for certain character traits. It goes into even like race genetics and whatnot, right? And but then one point Stephen Jay Gould makes very clearly, and in a couple of his books, but certainly in a lot of his public work, is this notion that it's about variation, and it's like you could have a certain variation. It could be completely contingent, and just depending on for some arbitrary reason in the given environment, that variation could survive over some other variation, but you can't plan uh, pre hoc or before the event really that these are you can't like, as in a crude way of putting it would be evolution. You can't. It's not really deterministic. You can't plan it in a way that you can write like some kind of algorithm and so to speak in that way. But I'm just curious. So in your kind of as a philosopher of biochemistry, how do you? I guess my question for you: what's your ontology of evolution? How do you understand evolution? Is it through this kind of Stephen Jay Gould framework, or do you take some other approach? Just if you could kind of expand on that. Sorry, I know
Yeah.
that was a lot there, but I too don't really know how to ask that question because it's like a lot there.
Yeah, no, this is a really a fascinating question. And when, like you said it before the interview, it gave me a lot to think about because I'm not sure that I have something like as a full ontology or philosophy of evolution, really. As for me, it represents a kind of like necessary context in which the things that I'm interested in operate. However, given that it is this kind of like necessary context in a way, or a context that I need to take into account, I think that it is an interesting question in itself. I think that the point that Stephen Jay Gould makes about the fact that we need to consider variation points towards something that I think is crucial for our understanding of evolution. That is that evolution is always an interplay of multiple characters, or there are multiple agents in evolution. And mostly we do have organisms or proto-organisms or monocellular organisms and the environment. Now, the environment is also made of other organisms. So the moment in which we make this distinction, of course, we are making an abstraction in a way. We are kind of separating things that in the world are really a constant interplay. But I think that it is crucial to understand that there is no, I think personally, that there is no objective progress in evolution in itself. Why? Because it is always a progress in a context. And I think that this is something that Gay Good underlines a lot. That is okay, like, you have improvement into for what? It does this like quite extensive discussion of like baseball. We might become better baseball players in the game of baseball, which is a game in which we selected the rules. We selected which parameters are going to be stable. Now, what it is quite fascinating about the world is that the world keeps on changing. And parameters change. I mean, now we are seeing it with climate change, really, that we can change environmental conditions in terms of temperature, amount of water types, how the water comes down. So whether we get storms or whether we get periods in which it doesn't rain for very long. And this affects the organisms. This might also affect how the organisms play, and organisms that were extraordinarily successful before might not be after, in the moment in which the context changes. So I think that the importance of the environment is really crucial, and asking ourselves, how do we want, like, I do find that in this context, progress evaluation is always to be contextualized as a progress for what, and progress in what, and in relation to what. So we need to make all these parameters. So what do we want to take out from our evaluation? Do we want to consider, how long we live? This surely has improved in time. Do we want to consider what is our reproductive power? And this has changed due to a lot of reasons, and this is fine. But I do think that indeed, like progression, or just the principle of the survival of the fittest as the only thing in evolution is a kind of, I don't think it leads us very far. I think that if we want to have a kind of better understanding of how evolution works, surely the notions of variation, the notion of environment, and the fact that we can't, I mean, we can predict the future in the sense that now we have good scientific models that give us statistical reasoning for which we can find that a scenario is going to be more plausible than another. However, it's very difficult to predict how each entity is going to react, or how each type of entity is going to react to those variations. So this is where I think that the levels of interactions are really important. Of course, other interesting questions, and in the project AssemblingLife I'm working on at the moment, we are really asking ourselves whether evolution is the only principle for complexity, for instance, and whether, for instance, variation, how do we want to consider the role of variation in those? And for us, is the question of whether some self-assembly or self-organization principles play also a role in bringing in change into the evolution, into the biological world. I do think that, very similar to what Gay Good would have said, that one of the main outputs of life is indeed for variations in complexity and simplicity, really. And without, and as he points out very clearly, without one of them being better than the other. Why? Because some work better in different environments than others.
Indeed, indeed. You certainly articulated that a lot better than I did. And the term you used, parameters, was quite important. I think I should have mentioned that. Also, I probably should have put the stipulation that yes, certainly with almost, I'm being dramatic here, almost infinite amount of compute power that we have. There are statistical models we could make, but the problem is they're probabilistic and they're models. It's kind of like predicting the economy. It's not something we can do because of how open the system is open in that sense. And yes, I'm glad you mentioned assembling life. Francisco, I have a whole bunch of questions just to discuss on self-assembling. However, before we get to that, I kind of want to kind of still stick to a bit of the metaphysics because that's what really interests me. I watched your TED talk on process philosophy. So, I mean, already reading your work, I've read three of your papers. And in all three of them, it's quite clear you do kind of talk a lot about processes and kind of this relational aspect. You don't say it explicitly, but I get that spirit in your writing in some sense. So if you could tell us, yeah, so again, researching in doing research in biochemistry, how has process philosophy helped your understand, not help, let's say, influenced your understanding of what are entities, what are things, and then more in your field, what are biochemical entities?
Yeah. So I have to say that I didn't start from a process philosophy approach. So I don't know whether this makes my case better, but
Who
uh,
knows? Some might
but
say it
maybe.
does.
Exactly, because I actually started thinking in very like neo-Aristotelian terms. So when I started working on these questions, I had a very like neo-Aristotelian metaphysics, which still influence parts of my thinking, especially in terms of kinds. But I did start from this kind.
Can you outline what that is, just for the listener, the Aristotelian what?
Yes, of course, of course. So, a Aristotelian metaphysics is mostly based on a view of the world as composed of substances and properties, or objects and properties. So, where substances are characterized by uh, permanent properties, intrinsic properties, and generally we can identify what a substance is because a substance resists to change. So substance is what remains through change. So for instance, the classic Aristotelian example, I can be seated or I can stand. What is it stable in the two states? Francesca is sitting and Francesca is standing. It's Francesca. So Francesca is a substance. Why? Because some properties remain stable. Um, this was more or less my view of the world, which is a pretty common intuitive view of the world. The moment, though, I started approaching, in particular, molecular genetics, which was really my first case study, I realized that if I would go in with this view of substances, substance view of the world, and see what genes are, I would have probably needed to give up on whether genes were anything because genes are extraordinarily dynamic entities. Um, so they are highly context dependent. So they depend on the fact that there is a cell that can transcribe them. The same gene in the sense of the same entity that encodes for a given product might actually be realized by different bits of DNA. So somehow genes were almost the opposite of the substance in the sense that they actually change through change. Now, what I ask myself is like, does this mean that there are no genes, that we need to like give it up, that there are no entities like that? And to me, that wasn't really a satisfactory answer because I did think that there is something there. And normally the principle that I use philosophically for this type of situations is that I ask whether the given entity has causal power or not. So does it impact the world around it or not? And somehow with this other kind of philosophical guidance, I was like, okay, maybe genes are things, but they're just things that are a bit different from substances. They are temporal entities, so they change through time. They might come in and out of existence. That might come in and out of existence because they are in a given context. And somehow this like became quite a framework in which I started thinking of biochemical entities more in general. And of course, being influenced actually mostly by the work of James Ladyman, simply because I was doing my PhD in Bristol. And a lot of my colleagues were working with ontic structural realism. So his work is mostly in philosophy of physics, but of course he underlines a lot that things are what they are because they are in given relationships. And then, of course, I got exposed to John Dupre's work, which again, Exeter, a bit further down south, but still same part of England. And I was like, okay, then there are alternatives. So that there are alternatives in philosophy if I want to still save some biochemical entities or molecular biology entities like genes. I don't need to stay with this neo-Aristotelian substance view for these type of entities. I can use these other tools. And then, of course, I don't deny that surely the fact that I've been exposed to quite an extensive history of philosophy training might have influenced me in the background. So at least gave me the idea that there might be different ways to think about to think about the world. And from that I really started noticing how actually how much things around us are actually contextual. And somehow, like the identification of substances is somehow always an abstraction. So we take things away from context. It can be very powerful. It can be really useful. We can do a lot of interesting science by kind of like taking away things from their context and trying to look at them in themselves. But the moment in which you consider them in the word, I find that they're always embedded in the context. And the more I started reading this, the more I saw that even in like political philosophy or social ontology, there is this underlying that like, I mean, thinking about, I don't know, gender and Celia's Langer, like even gender can be defined completely in terms of social relations. And then you think about entities like money, and then you talk to the philosophers of physics, and they tell you that even particles are really defined in terms of their interactions, and somehow this comes through everything.
Yeah, so do you still take a relational approach towards reality, kind of the Whitehead sense, or yeah, where do you stand in that regard?
Yeah. So I don't know how much I want to extend this view in the sense that I feel that I have grounds to do that for the case studies that I've studied. But I haven't studied the whole world as a system, so I probably would need to think about it a bit more. But in general, I find that it is at least an interesting heuristic. So I think that it is at least an interesting starting point, and I think that for my colleagues or for all of us that want to think about these questions, I think it is good to be exposed to the possibility that things aren't substances, that things aren't self-standing individuals. And then maybe like the result is that in some cases we are, in others think other things are not, or it might be that in some cases it is very fruitful to think in terms of substances, and in others it is not. But I think that in principle it is good to have the option um, as a possible heuristic.
Yeah. I mean, again, I see, you're a true academic. You really try to take a step back and not ever make this all-encompassing claim. But this is just a
Yeah.
podcast, so you can speculate as much as you want. There's no that's the fun thing about a podcast. It's just a podcast. There's nothing, it's gonna. Well, apart from the brutal comment section, no one's gonna hold you accountable for what you say. So,
Yeah,
so,
yeah,
but.
that's fair enough.
Francisco, I'm glad you mentioned the gene. I mean, I think it's interesting. I found you through your paper on emergence, but then I'd say the paper I like the most is the one on the gene, because like yourself, I too have been fascinated by, yeah, what's the gene? I remember when I was like 15 or 16, I read Richard Dawkins's famous book, The Selfish Gene, and so, but I want to have a whole part just to discuss that paper. So for now, let's shelve that because I want to stick to this notion of self-assembling processes and kind of along these lines. Um, but yeah, again, a very naive question. Could you explain to us? So as you did say, you're working in the Assembling Life project at the University of Oslo, and could you tell us at a naive level? what is a self-assembling process? How can we understand this as suppose if you could use some phenomena in the world, both biological and probably even social, to explain this to us?
Yeah. So firstly, the definition of self-assembly is extraordinarily controversial. So for all of us, the fascinating aspect of the process of the project, and I imagine why it got funded, is because while the notion of self-assembly as the idea of like things assembling each other together is kind of intuitive, the characterization of the phenomena is extraordinarily complicated. But of course, there are some mapping definitions, or there are some starting point definitions for which we can define self-assembly process as the formation of an ordered structure from parts that are not equivalent, so they are not the same, and that move towards a form of equilibrium. Now, classic examples in the disciplines are forms of bird flocking. So when we see the beautiful birds presenting, like, for instance, a triangular shape in the sky, that's a form of self-assembly. Why? Because the individual birds move themselves into this ordered structure, and they do that. Why? Because they can fly more efficiently. So they create a form of equilibrium for which they use less energy. And they have, of course, like this fascinating then loops in which the birds move down the line so that somehow the total amount of effort that the bird flock puts into flying reaches a kind of optimal or suboptimal or closer to a better one. Looking at other cases, protein folding is also another case that is mentioned as a self-assembly process. Why? Because we start from this linear but complex chain of amino acids. So again, non-equivalent parts, and these parts fold, or rather fold up into a globular shape. And again, they do that on the one hand, they reach a form of equilibrium, so the folded structure of the protein is more stable. But then it's also interesting because the folded shape is important for functionality. So, and this brings in further considerations. Another important notion that comes with the notion of self-assembly is the notion of self-organization. Now, again, the distinction between self-organization and self-assembly is really debated, but if we want to point out some differences, is that we still, also in self-organization, we go towards a form of ordered structure, or they even say asymmetric structure, but we start from a population that is homogeneous. So we start from things that are the same, and they characterize themselves in order to reach a new level of order, and this normally takes a high amount of energy. The classic example for this is embryogenesis, so in which we start from a rather simple entity, and then the cell starts specializing themselves. And we reach a form of order, or a final form of order, which is a finite organism that needs a lot of energy to maintain its status, because we need food and there is metabolism and so on. But we started from a rather homogeneous population or homogeneous sets of entities. However, these are just some working definitions that we are working with, and I thought that for the purposes of the podcast, indeed, they can allow us to map the differences between the two. But there is so much work on this, and individual scientists use these notions in different ways, and this is probably also why it is so fascinating.
Yeah, I mean, the way I think of it is that this assembling takes place in some collective, let's say, if that's a proper term, without any external direction or influence, right? I mean,
Yeah.
so I mean, okay, all the examples, which is fair, you are a philosopher of biochemistry, you used were from biology and chemistry. So yeah, is there phenomena, let's say, non-sentient, non-biological beings, like could you say maybe I'm reaching here, but like the economy in some sense is a self-assembling process. Not that the economy leads to order, but I mean, can we see it even in the social world?
Yeah. So I do think that surely the notion of self-assembly, probably also the notion of self-organization can be applied to other cases. Our PhD researcher Sebastian Oster-Ost, he works on like various definitions of self-assembly, and he was telling us that the notion of self-assembly itself comes from mechanical engineering. So definitely something that is not um, sentient in a way. And we do have collaborators that work on, for instance, materials that display memory. So I know some types of metal, if exposed to heat, tend to take the shape that they had before. So there is the very cool example of the paper pins that if we open them and then we heat them up, then they take the clip, the original clip shape again. And this can be a case of self-assembly maybe, in which we take the material we expose it, of course, to some heat in a way, but somehow it seems to take the shape again in itself, in the sense of like without functionality. And I think that probably the notion can be useful and can be used in a useful way to describe like also maybe like, yeah, societal organizations or economies. Like something that we were discussing recently is whether indeed it is these are cases of like model borrowing in which we have these general models that can describe self-assembly, and then they get borrowed by different fields and might lead to different results. Then, of course, the question for a philosopher is, are we really doing the same thing, or are we talking about phenomena that are different, but we have this tool that is quite useful, so we use it in both ways? So are all these cases genuine cases of self-assembly, or are we just using a useful notion because it works and get a yeah and get a good result? I am more inclined towards this second. So if this is just a podcast, then I need to have a strong opinion.
Yeah.
I would say that, yeah, probably they are not all self-assembly in the same way. So we can use the notion to characterize them successfully.
Yeah,
Uh,
this is the problem with definitions because like you once at a certain point, if they encompass everything, they become meaningless. Because it's funny, my background's in computer science, right? Like, and computer science
yeah.
it's highly related with systems theory, and we even have this notion of like self-healing in networks. So in like peer-to-peer networks, the way kind of modern systems work is like a lot of it is it's distributed systems. And so you have like, let's say, 5 servers, and if one server crashes, how can this network still be live? And I remember we used to learn about this notion of it being self-assembling. But the thing though is a lot of that is still directed, as in it's the foundational algorithms, the set of rules are set by engineers. So you can't really
Yeah.
call that a self-assembling process, the same way you would for a biological entity because it's a different category really. So
Yeah.
I find that again this is the problem with definitions as Wittgenstein pointed out very well.
Yeah.
Yeah, back to the project. Just I don't think we can go too deep into this just in the interest of time, but if you could give us just a bit of a tease as to what kind of research are you currently doing in the AssemblingLife process, like what kind of questions are you asking and what kind of problems are you tackling?
Yeah. Yeah. So, well, there is the most general question which we just discussed.
What you-
That is, how can we characterize self-assembly and self-organization? And what are the historical roots of it? So, indeed, like our PhD researcher is also working on doing an historical analysis. He's not an historian, he's a philosopher, but this is quite useful for us. Then we have our PI that is mostly working into whether self-assembly explanation can be different from, for instance, genetic explanations. And this, again, is quite an interesting one, I think, also in relation to what we were just discussing and to what you just mentioned as why you use self-assembling computer science. Because, for instance, it seems to be that in the case of genetic explanation, despite the fact that, of course, there are genetic networks and it is quite complex, but you kind of start from a thing that is maybe similar to a code, and then you play in a way. So you start from these sets of rules, if you want to call them in this way, and then you can infer a series of things. While maybe in self-assembly there is something else going on, because you do indeed get this diversity, and somehow the way in which things interact leads you to an ordered structure. So there is a question into whether like genetic explanations are different from self-assembly explanation. We are currently discussing the work of Kaufman in this regard. And this links back to what we were saying earlier, like how many forces are there for life? Is it just evolution, or do we need something else? So another question is there a principle of self-assembly and self-organization that was there at the beginning, so at the origin of life, and that allowed the whole evolutionary dynamic to start, or that cooperated with the evolutionary dynamic? And this is very related to the study of the complexity of the systems. And then we have my colleague, the other postdoc working with the project, which is Bendik Allemabi, and is working on notions of self-organization in relation to intentionality. So group self-organization, animal behavior, so the case of the bird flocking, for instance, that I mentioned earlier. So we kind of try and look at these problems or at these questions from another angle. And we had a research assistant as well, that is Ye Tong Hu, and she has done a fantastic work on protein folding. So this has been quite a specific, narrow case study, but she looked at which types of causal relations are going on during protein folding, which type of explanations we can use. And somehow it seems that a mechanistic view of protein folding wouldn't deliver us the results that we want to have. So you see, the project is quite broad. There are also questions regarding whether self-assembly can challenge our understanding of emergence, for instance, and relations like kinds and classifications and other questions.
Yeah. What do you mean by your colleague who's working on intentionality apropos self-assembly work? Can you kind of expand on that a bit, please?
Yeah, so basically, like, I think that what is mostly starting to look at, because it really just started, is the, like, for instance, in the case of bird flocking, like how much intentionality is there in the birds informing the flock, and how can we explain collective behavior? Do we need to postulate a kind of like active intentionality of like, okay, now we want to act as a collective flock of bird, and we and we take the sheep, or is it really a kind of like energy optimization? And so somehow with this almost like self-assembly wave,
Yeah.
we bring things together.
Yeah, the reason I asked that is because like for me, whenever I hear words like intentionality, I think of agency.
Yeah.
And I've always, maybe I'm wrong here, but I've always viewed the notion of self-assembly kind of as it's more of a phenomena without an agency. It kind of, just kind of it emerges without any specific agent. Then the question is how you define an agent. So that's a whole other
Sure.
door to go, something to explore. But okay, excellent. So that makes sense, and that's why I used the birds as an example. Excellent. So just on the notion of emergence, I think just again in the interest of time, I do want to touch on the gene before we get into emergence. And if we have time, we can get into the emergence too. But Yeah, this was an excellent paper. I really enjoyed reading it. And yeah, I think I took down too many quotes, and now it's even confusing me because I should have tried to, compress them a bit more to make them into more of questions. Um, but yeah, this paper, the gene as a natural kind. So firstly, before we even speak of the gene, what in the philosophy of science is this concept, natural kind? What does that mean?
Yeah, yeah, excellent question. So, again, as always in philosophy, there is no univocal understanding of this, but I think that everyone would be happy if I say that a natural kind, it's a general category that captures real features of the world. So a way to talk about it, taking it from Plato directly, is that it is a category that carves nature at the joins. So we get some, let us consider a kind of univocal natural kind, gold. Gold is a natural kind. Why? Because it captures all those instances of gold that are in nature, and it tells us that instances of golds are going to have a specific atomic number and specific properties. And this leads into a second important aspect of the characterization of natural kinds, that is that natural kinds are categories that should be projectable. So we should be able to infer the properties from the general category to its instances and from one instance to another. So if I know that this instance is a gold molecule and this instance is a gold molecule, then I can project the fact that they're going to have similar properties and similar behaviors because they are classified in the same way. And also, natural kinds are nodes in causal network. And here I'm really using Mohammed Ali Khalidi definition of natural kind. And that means that these entities need to play a causal role in the world, and the properties needs to be causally related. And so this is the approach actually of Khalidi, but I think that a lot of philosophers would be happy with conceding that natural kinds need to be projectable, they need to be useful in science, so they need to allow us to make generalizable, successful claims in terms of predictions and explanation, and they need to have this causal role.
Yeah, excellent. I mean, in fact, while you're talking, when I'm editing this, I'll put the quote you've outlined. You enumerated ABC as you mentioned, so I'll put that quote just to make it easy for the
Super. Thank you.
viewer. Yeah, excellent. Okay, now the gene. It's okay. This is my own ignorance, Francesca. I always thought that biologists would have thought of the gene as having some kind of natural kind. I didn't think that it was quite controversial and, as you say, has been challenged by post-genomic discoveries. So, yeah, could you please firstly give us a bit of a historical account of after the post-genomic discoveries, how this notion of the gene being a natural kind was challenged?
Yeah.
And then we'll get to what you propose in this paper.
Absolutely. So, for those of you that don't know what we mean with like post-genomic discoveries, we means basically the discoveries that we made thanks to the Human Genome Project mostly, and thanks to the 3 waves of ENCODE, which is this fantastic project of trying to map basically each genetic sequence or each relevant genetic sequence with its functionality. And code has happened in 3 ways. The last one finished quite recently, and we keep on working on, or rather I don't really, but they keep on working on trying to map genetic sequences with functionality. Now, what we found with these waves of quite varied research projects is that the link between the DNA or the RNA for some species, but let us stay with the DNA for simplicity reasons. The DNA and given products or given functionality, the link between these two things is way more complex than we thought. So the idea, like let us say in the 80s, is that, okay, fantastic. We discovered the DNA. We know that the DNA is composed of genes. Genes are sequences, like the classic definition is that genes are just stretches of DNA that encode for proteins. what do we need to do to understand the whole complexity of biological beings, given that this is dependent on genes, is that we just need to have the whole DNA sequence. If we have the whole DNA sequence exactly as a program for computers, we're just going to get the whole complexity of human beings. Why? Because we just find out how many genes we have, where they are, what do they do, and we're kind of sorted.
You can throw, I mean, that's a really, I'm glad you mentioned that because I think colloquially, people say things like, I can't help it, it's in my genes. It's this very essentialist notion. So, I mean, I think a lot of people still think of genes in this very kind of essentialist way where it's like almost like a program, and then that's it. All the phenotypes and everything is that's it. Anyway, I thought I should
Yeah,
mention.
and absolutely. And I mean, in kind of like defense of all of us that thought in this way, or that keep on thinking is in this way, a lot of books and handbooks in molecular biology, not only for high school or like primary education, but also for first years of undergraduate education, keep on defining genes in this way. Like genes are contiguous stretches of DNA that encode for a product, which is actually quite true and works quite well for very simple entities like bacteria. If you're studying bacteria, you can kind of get this quite simple picture, even if, of course, they are also very complex and they display complex behaviors. But for humans, or for more complex eukaryotes, the situation is way more complicated. Why? Because the levels of interactions, and this is what comes through with the study of like genetic networks, for instance, which is a model to understand such complexity, epigenetics, the role of the cell in reading the genome. Like what we found out basically with the Human Genome Project is that, okay, it's not enough for us to have the whole DNA to understand what a human is. And not only that, but we found out that the same stretches of DNA can play different role in different environments. So according to how the cell reads the DNA to keep this kind of like almost coding language, the way in which the cell reads the DNA changes the type of proteins that is going to be produced or the type of molecular products. That according to environmental pressures or needs of the cell is able to read different bits of DNA at the same time in order to give a given product. So for instance, this completely challenges the notion of the genes as contiguous because they can actually come through different bits in different moments. And so somehow what we found out is that, well, the gene is not a contiguous stretch of DNA anymore. And this is how the question of whether genes are even a thing anymore came true. And this is how the question of whether genes are a natural kind came true. Why? Because this definition doesn't hold. And this relates to what we were saying a bit earlier, you know, that like if you come in with this quite strict view of the world in which, again, you want to have a substance and the gene is a substance and it is this contiguous stretch of DNA which is permanent and stable and is going to play the same role in all contexts. And so once you get that information, you get a given product always. This notion crumbled. And even if you take that notion, then there is no gene because or rather maybe there are some in some simple cases or simpler cases. But you can't generalize that to everything. Or the amount of genes that you have is extraordinarily narrow, is much narrower to the one that would explain the complexity, for instance. So there is the case of, for instance, cystic fibrosis. There we know that there is a single gene that is going to cause the disease, and somehow we are very lucky because in that case, it is something that we can really target. Yeah, of course, it is a problem for those that suffer from the disease, but from a research perspective, it's a kind of easier target. But a lot of other even diseases or traits are polygenetic, which means that they are caused by different bits at the same time. And so again, like my question is like, okay, we do so much stuff still referring to genes, and genes do play a causal role because we get proteins. It's not that we don't get them. So there is a transcription, the cell is doing stuff. But this notion of genes is not working. So how can we save the genes in a way without by embracing this complexity? And this was basically what led me to write both the paper on the emergence of the postgenomic gene and the paper on the gene as a natural kind is basically this question. Yes.
Paper, you do argue that the gene is a natural kind. So, I mean, that's the part I find quite interesting. I mean, if you could expand on that, as an
Yeah,
after we've developed this, you've given us this history. Now, kind of the
yeah.
argument that you make here.
Yeah. So basically, the argument that I make is that, okay, the gene can still be seen as a natural kind, is a natural kind, while because if we take a definition of gene as those bits of DNA or nucleic acids in general, RNA for some species, that encode for a given functional product, which can be a functioning RNA or a protein, if we take this definition and we take away instead the rigidity, if we take away the fact that it has to be working in a kind of self-standing way, and we embrace the fact that these entities come through during transcription, then we can apply those definition of natural kind that I mentioned, that the definition of natural kind that I mentioned earlier, that is, okay, if we take this definition of genes, if we characterize it in this way, then we can make generalization. We can, the kind is projectable. We can infer that from one instance to another, that gene is going to display different properties. Why? Because what we keep stable is the production of the functional product instead of the sequence. And then with that, we also we can also have causal power. So the genes have a causal role. So in this way, they are a natural kind. Now, what is quite important in my definition is that you need to have a specific realization for the genes. So it's not that anything can do anything. It's not a purely functional notion of the gene. So you need bits of nucleic acids that realize the gene. And I think that biologists are very good at telling us which of them. But we need to get rid of somehow this reductive constraining and rigid rigid notion in order to get to the fact that the genes are a natural kind. And then if we embrace the complexity and the context dependency, and we take this functional definition, then I think that genes are still natural kinds.
Okay, so you still would contend that there's like this. I mean, for me, it seems like when I was reading this paper, there's this tension between the fact that, well, they are a natural kind, but they're also highly context dependent. But then, as per the definition you gave from philosophy of science, you can still have kindhood or kindness, despite it being highly context dependent. That's kind of the point you're trying to make here, isn't it? That
Yeah.
tension is sort of, it's there, but it's not. That does not make a gene not a natural kind. Okay, okay. I mean, okay. Now again, to get a bit speculative here, because this is a podcast after all. I mean,
Good.
sure you're familiar with, the big name I can think of is Dennis Noble, who's been very critical of this kind of Richard Dawkins selfish gene approach where you reduce all of life to the gene. And then this recently, about a year ago, I've been trying to read his papers, but they're very technical, and for a neophyte like myself, it's really hard. This Argentinian biologist called Alfonso Martinez Arias. I'm not sure if you're familiar with him, but he's written some work on just like, yeah, I think he takes more of the Stephen Jay Gould approach about variation and whatnot, but he's also very critical of this like reducing life to a gene. And but saying the gene is a natural kind, you're not saying ipso facto that through that you can just say all of life can, when I mean life, I mean let's say human life, uh, can be explained purely by our genes, correct?
No, absolutely not.
Yeah, yeah, yeah.
Absolutely not. The point is simply to make that there are things in the world that are genes. They are more complex than what we thought they would be, and they need
Yeah.
to be characterized differently, but they are still there. And if anything, the view that comes through is that, I mean, here I'm really quoting Carola Stotz and Paul Griffiths, that genes are what cells do with their genome. And I
Oh,
think that And I think that they capture perfectly and they influence my work a lot. And the work of Carola Stotz in particular, like she influenced my work a lot on how we need to think of the genes. That is, while they are not the things to which we can reduce everything, they're kind of instruments that cells use for their functionality. Now, this leads us a little bit to whether there was before the chicken or the egg, and and questions of ontological priority, which
Correct. I was
I
just
think are really
gonna say,
fascinating but complex.
yeah.
Um,
This is the kind of the notion of like, I mean, I don't know, because you said genes are something that exists in the world. Are you saying therefore you're a scientific realist, or what is it, what is that notion of existing in the world it really means, right? I mean, those are again a whole other more like metaphysical questions, which I don't think we'll have the time to get into here. This again, we've got about 15-20 minutes. I hope that's okay, Francesca.
yeah.
So this is probably going to be the penultimate question. It's funny. A result of doing this podcast is that I always I research on the guests, person like yourself, and then I think to myself, what the hell am I doing with my life? It's just because I gotta say congratulations on the fact that you're working on this monograph, which is the philosophy of biochemistry, biochemical kinds and their properties. I'll put an overlay for the viewers if you're watching with the chapters you'll be covering.
Yeah.
And yeah, I mean, this is an unbelievable project. I mean, it's a complete monograph with the Cambridge University Press. So just, yeah, give us a bit of a teaser as to, yeah, there's even a chapter on AI and biosynthesis, certainly a very pertinent topic for our times. Yeah, just, I mean, we did discuss a lot about biochemistry, but is there anything that you want to bring up that you'll be covering in this book, apart from some of the topics we discussed so far?
Yeah. So, I mean, the purpose of the book is really to get a discussion going. So that's why I was motivated to do it, because philosophy of biochemistry is still quite narrow. I do think that there are a lot of philosophers that work on questions that would be within the philosophy of biochemistry, but they come from biology, chemistry, philosophy of science, philosophy of genetics, and there is no kind of like systematic discussion yet. And so the idea of the book is that, I mean, the book is less ambitious than it seems, in the sense that it's like, okay, we have these biochemical kinds that biochemistry is talking about. Questions of classifications are relevant. What are the properties that pertain to biochemical kinds? And then the book wants to go through three that I think are the main properties, that is chemical structure. So how do we need to think in terms of structure when we talk about biochemical kinds? And I think that their proteins offer quite a challenging notion, because proteins have a structure that is very complex, that is dynamic, is characterized by self-assembly, as we mentioned earlier, and this is quite different from gold. Gold is like, I mean, also elements are dynamic in itself, and we know that structure is an abstraction and everything, but the complexity that we find for proteins is quite novel.
For sure.
Then there is a big chunk on biochemical functions, which I find mostly fascinating, and this will be based on what we discussed together and my previous work. Then there will be a chapter on evolutionary properties. And this, again, relates to some of the things that we were saying in relation to, okay, like, can molecules evolve? What's the role that molecules can have in evolution? And this is quite a fascinating chapter. It's also one with which I am struggling a bit, because I think that the questions are really difficult and they pertain to astrobiology, the origin of life question. The chapter on the relations between these properties will kind of be based on my previous work. So I will argue that there is a form of weak emergence for biochemical kinds. But again, like, given that the book wants to get the discussion going, while I will put forward a view of biochemical kinds, that is, they are characterized by structure, function, and evolution, and they are weakly emergent, I'm also going to kind of leave the discussion open at the same time. So I want also to give the reader an overview of the options that there are, that you can be a reductionist or you can be an anti-reductionist. And this is because the questions really need to be discussed still. So I will try to bring that aspect forward. And then, the very last chapter is going to be fascinating. I need to say that when they gave the Nobel Prize for chemistry this year, I was really overexcited because I was like, well, this is really a Nobel Prize on an AI tool that studies protein structure. And why does it study protein structure? Why is protein structure interesting? Because it plays a function. Um, so yeah, I'm quite excited about that. I do have some ideas into how I want to approach into how I want to approach the application of AI in these cases, which is mostly as an instrument, so as a scientific instrument, so picking up on other literature on
So
this.
wait, I was mistaken. Was the Nobel Prize in chemistry also given to some work done in? Oh, I didn't know that because I thought you mentioned you were talking about the Nobel Prize in physics, which was given to Jeffrey
Oh,
Hinton, who's an AI
yeah,
researcher. So even in chemistry, excellent. So it was given
yeah.
for the protein
So
folding problem, or
yeah, yeah, it was given for the protein folding problem and to the teams that worked on Rosetta,
yeah,
which is one of the first.
that was a
Yeah,
brilliant
yeah,
discovery.
exactly, and on AlphaFold. So that was also interesting because they gave the Nobel Prize to researchers that are not working for universities, but
Yes.
for Google DeepMind. So that also caused its own. um, but yes, so it was given for the which for us that work on these things was, I mean, for me, I was really super excited.
Yeah, I like
Um,
the approach you take. I mean, you're right. I mean, in fact, the initial email I sent you probably remember, Francesca. You were the
yeah,
first person that I came across who was a philosopher of biochemistry. That it's a
yeah,
very niche, you could say, but it's certainly a burgeoning field. So, a book like this, for those interested, is certainly for the ilk, with that ilk, they could yeah, kind of asking the right questions. And I think that's what philosophy does best, isn't it? It's more giving concepts and reformulating what we think we already know.
yeah,
Because
yeah,
no, I was going to ask this question from you at the beginning of our conversation, but maybe now, given we're discussing your upcoming book, is a good time to ask this. So all of these questions you're exploring, or all of these ideas you're putting forward, the theories you're putting forward, why aren't why aren't biochemists just putting these forward? Like, as in, why do we need a philosopher to step in to
yeah.
put forward these ideas and ask these questions?
Yeah, yeah, fantastic. Well, this is a thing that we discuss a lot in philosophy of science in general. And my answer is simply that, well, knowledge is a collective enterprise, so we need to stop thinking of like knowledge of science or philosophy even as something that is done by an individual in its own like little lab or basement flat or fantastic studio flat, and just thinking and just doing experiments. But knowledge is collective, and I think that it is important to divide labor. Like, I do think that biochemists are doing a fantastic job at doing all the lab work and the kind of like hands in the field work, but that work requires time and energies. I have friends that work in molecular biology that spend weekends and like 15 hours a day just running the experiments. Um, and at the same time, thinking requires time and requires space and requires a level of abstraction or distance from the problems. And this is why I think that we really need to cooperate, because I don't think that anyone really has the time to do everything. So it's in a way simply like, well, we are both interested into these questions of for instance, how structure relates to function. Last year, I had a fantastic conversation with Andrew Lowering at the University of Birmingham, who is a biochemist working on protein structure and function. And we're like, okay, we both have this fascination, like, what is the function of proteins? And his lab and his team were running fantastic experiments on these, but they were spending hours and days just trying to understand how the model works, just trying to write down the program, then going into the lab. And at the same time, I have the same questions, but instead I have time to go and read about the history of biochemistry, for instance. And I have the time to read about different definitions of functions and give an overview of the literature and say, okay, we can talk of functions in these 4, 5, 6 different ways. And also of structure, like, do you mean primary structure, secondary structure, tertiary structure, quaternary structure? And of course, it is not that they don't know what they mean. It's just that they are so, not all of them, but in general, I just find that it is such a taxing job to also run the experiments that I think it is good to have someone that instead is just thinking,
Yes.
because somehow it can provide, as you said, I think that philosophers provide good questions, but they also provide good conceptual clarity.
Yes.
So,
I
so
really like this I think it was in Wittgenstein's biography that by Ray Monk. He kind of subverts that. He says that it's not only that philosophers, which Wittgenstein says is, it's not only that philosophers give conceptual clarity, but uh, they also kind of reformulate the whole epistemic space. And what he means by that is like, okay, you may think that a function or that a self-assembling process is this thing. You may start with that assumption, but a philosopher can introduce a concept that makes you, rethink all of the assumptions you had made beforehand. And I think that's as before we started recording, discussing Kant and all of that. I think that's where it gets quite interesting. And I completely concur. I mean, this question that I asked from it is kind of almost yeah, you're right. It's almost an overused cliche question, especially discussing philosophy of science. So I didn't want to spend too much time discussing that because you're right. A kind of labor of thinking. It's a collective discipline. It has to be done in a community, and I see this a lot in my field of computer science and AI for sure. Okay, Francesca, on the note of philosophy, since we're running out of time, I thought why not just end with a beautiful, romantic, metaphysical question. Given you're working in biology and chemistry, what's your philosophy of nature? How do you view nature? What is nature for you?
oh, yeah, wow, this is like a massive big question.
I thought we could start. We just end with something beautiful,
Uh,
romantic.
yeah, I mean, if I do, so given that you encouraged me to just like be open and say what I think, like just on the wave, I would almost say that nature is everything. So in this sense, I do have this notion for which, yeah, like we keep on as humans, like separate ourselves from nature as if we are different, and maybe we do that because it is convenient to study, to use nature. But I actually think that we are part of this bigger thing that is nature, and even things that we would consider are artificial, like I mean houses, cars, our computer, like they're kind of a product of our interactions with the environment, and we are not unnatural. So in a way, I do think that we are all part of this big thing um, that is nature. So if I would need to, yeah, if I would need to define it, it would be that. It would encompass many different types of entities. And I do think that other important features of it are the fact that nature is extraordinarily varied. So variety and complexity is its main characteristics at the moment in which we try to find commonalities. It's possible, but we kind of take away the richness. And the other bit is that yeah, nature is based on interactions. So this leads back to what we were saying, no, this constant context dependency that each component of nature has. So I am, as a kind of like, yeah, summary, like I think that things are what they are because of the relationships they are in, and this whole big set of relations kind of
Yeah,
constitutes
yeah.
nature.
Yeah, and I like that you said that nature, yes, nature is not a monolith. There's even a Hegelian philosopher who says that in some sense nature doesn't exist. Nature is an abstraction. But nature,
Yeah.
if you look at the world, it's too complicated to just abstract out like that.
Yeah.
Excellent. I thoroughly enjoyed this conversation, Francisca, and thank you very much for being generous with your time and especially your knowledge. Yeah, thank you very much.
Well, no, thank you so much. It has been fantastic. And I mean, philosophy is my greatest passion. So I just love to share it in all ways.
And certainly, hopefully, we can chat again once your book is out, because maybe I could read it and ask questions pertaining a bit more
Yeah.
to kind of the chapters outlined in the book.
Yeah. No, that would be fantastic. Thank you.